Hello, and thank you for taking the time out of your day to listen to my presentation here at the Peace Conference. I know we were all hoping we'd be in person, but rather, once again, virtual. Today my talk is going to be in and around the Leap-In transposase platform, and specifically how we've started to think about applying this platform to create CHO cell lines for the manufacturing of multi-chain bispecifics. And I think at the end of the day, what I'd like you to take home is that the pools derived from this platform, the initial selected pools, actually have a unique capability of enabling the manufacturing of these biologics. And unlike most presentations, actually, I'm going to spend the first half of my presentation roughly talking about things I don't have time to talk about, unfortunately, but let's see how it goes. What I don't have time to talk about, and is a presentation in and of itself, is the GPS platform that we have developed at Atom. Suffice it to say, we have nearly 2 decades of experience with design of experiments, identifying variables and feature sets within a biological design space, our ability to build these variants, test them, use machine learning algorithms to model them, and to feed this back into a loop of the design, build, test, learn cycle, which enables us to engineer biological functions, modalities, and phenotypes. Derived from this, there are 3 main pillars of this GPS platform. One is the gene GPS platform, which, loosely speaking, is applied to codon optimization. That is a presentation in and of itself. The vector GPS platform, again, thinking about those vector elements and understanding vector elements as they apply to the expression of proteins. Again, a separate presentation in and of itself. And of course, protein GPS, where we engineer proteins with novel fit-for-purpose functionalities and characteristics that maybe Mother Nature had not originally thought to design them to do. And again, the subject of multiple presentations in and of itself. But what I'm focusing on today, of course, is the Leap-In transposase platform. And the reason I bring up these other pillars is to make sure that it's clear that they are all part of the Leap-In transposase platform. The enzymes have been engineered using the protein GPS methodology to be fit for purpose. And of course, the transposons themselves, the DNA elements, are heavily influenced by gene GPS and vector GPS. And another thing which I'm not going to have time to go through, and I apologize, quite busy slide, is what a transposon and a transposase is. You know, Mother Nature has used this as a genetic engineering tool herself over billions of years, the subject of a Nobel Prize. But I think at the end of the day, the take-home message is that the enzymes moves DNA from one place to another, and it's very, very good at doing that. And that's what I'd like you to focus on as we start to think about the original selected pools in a CHO cell line, that the integration between these inverted terminal repeats that's facilitated by this enzyme is nearly perfect. And I also won't have time to talk about the process and the method by which we've applied this to biotech and taking expression cassettes for biological molecules of interest, potential therapeutics, and other, and applied the transposase platform to that in a process. But again, what I'd like you to take away, and from this slide, albeit busy, is that the structural integrity of the transposon, that element in between the ITRs, is perfectly maintained. Design it in silico, build it in the lab, it goes that way into the genome. And another thing I'm not going to have time to talk about is our cell line development process for biologics and for CHO cell lines. Again, we've highly optimized a process where we're looking at approximately 3 months from transfecting cells to having a research cell bank ready to be delivered to a CDMO or for master cell banking, et cetera. And again, I'm not going to go into the details here of all the processes here and how efficient it is and how robust these clones are. Rather, I'm going to focus on the pools that we're able to generate as part of this process. And so fundamentally then, what is it about these pools that makes the process more robust, that makes our pools uniquely enabling in some way, which hopefully I can convince you of in the next few minutes? Well, Fundamentally, it's about the integration mechanism. If you think about an expression cassette, let's make it simple. Let's make it an antibody with a heavy chain and a light chain, some regulatory elements, promoters, etc. around that, a selection cassette, in this case GS. If you put this into a CHO cell and you ask for it to randomly integrate this transgene, of course you will get the transgene of interest, that which was designed, that which was synthesized. Some of it will end up in the genome that way. But as we're all very well aware and has been published heavily over decades, that is not all that ends up in the genome. The pool of selected CHO cell lines have truncations, have deletions, have concatenation and scrambling. This results in a pool that is not uniform and as such, not very predictive of the final clones and not very useful in general for making too many decisions, especially when it comes to complex molecules. Of course, the solution to that from our perspective is to add ITRs, call it a transposon, add the leap-in transposase enzyme, and now what you get, as I've referred to previously, is perfect high fidelity integration of multiple transgenes across the genome. And importantly, not only do we have multiple integrations that are perfect, but each one behaves very similar to the next. And by virtue of that, we get a highly uniform pool of clones that comes out of that initial selection. Let me give you some examples of what I mean by uniformity in these pools. In this case, I'll be looking at titer of pools, and again, I don't have time to go into the details, but suffice it to say at this level, randomly selected clones here, looking at spin tube titers, we see in this particular pool of clones, 62% of the clones are in the top quartile of expressors. Right? 82% in the top half. If you compare that to a random integration-based methodology, high expressing clones are actually quite rare. The uniformity is here down here at the bottom where you have the truncations and the deletions and the scrambling. And instead of having to screen 5,000 clones to find a rare event, our pools are so uniform, and in this case, based on titer, that we can find those high titer clones in clonal cell lines by screening under 200 clones and in many cases less than 100 clones. This is an example of titer, and because we get so much integration and they're highly uniform in their capability of expressing the transgenes, this also means that if you have a good transfection efficiency, you should have quite rapid pool selection as well. And again, it comes out of the fact that we have perfect integration, we have uniformity in our pools, and so we can actually, in this case, 11 different antibodies. Yes, there's molecule-specific differences, but with a particular vector, we can get selection of these pools in approximately 1 week. With other vectors, it's even faster. Again, thinking about those characteristics of pools that can result when you have such efficient integration and you have uniformity of your integration, creating relatively uniform pools. In addition to that, I mentioned titer before, that we have uniformity of clones as we relate to the titer of the pool, but this is across pools as well. The titers of these pools that we generate are quite high. Again, high transfection efficiency, high integration efficiency creates uniformity, and so therefore if you don't have outliers, your pool should be reasonably high titer. And here we can see antibodies ranging from 5 to 7, almost 8 grams per liter, some Fc fusion molecule data. And what I'll focus on a little bit later, we've been spending more and more time on the 3-chain bispecific problem, and now we're starting to obtain pools in the 7 gram per liter range in the case of 3-chain bispecifics. This uniformity is also not just with respect to the titer, and I'll come back to that in a moment. But here again, in a case study that is a presentation in and of itself, one of our licensees, Janssen, has been quite verbal about and quite open about how they use our technology. And again, what I want to impress upon the group here today is that the pool titer will predict robustly what's going to happen in the future with clones. If we stay in the shake flask, pool titer equal clone titer, the pool must have been quite homogeneous. You switch the platform, you look at a phase 1 scale down model, none of the clones are any worse Again, Pool predicting a scale down model, and some of the clones are in fact even better, not surprising. In this case, going from 4.5 to nearly 8 grams per liter, and here for a hard to express molecule, again in the shake flask pools predicting clone titers, and then again in the scale down model, none of the clones any worse than they were in shake flask and many better, and in this case for a quote hard to express molecule 6 grams per liter. As I mentioned, this uniformity is not just with respect to titer, but also with respect to product quality. If the clonal product quality matches the pools must have been quite uniform. And here you can see a data set where in fact we see significant amount of uniformity of the clones with respect to their comparison to the pools, both for glycans and for charge variants. And again, another presentation in and of itself. So you take all of that, you mash it together, you add a little COVID, and here's a case study, which again, I won't go into detail on, but again, gives credence and precedence to the notion that the pools that we can generate are quite powerful and quite unique. In this case study, we were able to, with 2 partners, go from the email of a sequence to shipping to a CDMO of banked pools in 38 days. The titer of these pools was around 5 grams per liter. The CDMO adapted an intensified fed batch, which gave them 12 to 14 grams per liter, and based on that data, filed an IND for the treatment of COVID-19. Again, a uniquely enabling situation by result of the uniformity and the capabilities that are created by leap-in transposase derived pools. That's the first half of the talk. Let me summarize. Again, that's the part of the talk where I'm not going to get a chance to talk about a lot of the data. But here's the take-home message. Expression construct integrity is maintained. What you design in silico, you synthesize in the lab, it goes into the genome. Each integration on average, yes, there's variability in Mother Nature, but on average is equally functional with respect to titer, the amount of protein, and with respect to product quality. And this combined results in this unique ability to rapidly generate robust and predictive pools. And again, colloquially referred to as the power of the pool. So that's kind of a background on the leap-in transposase system and how we've applied it in multiple cases, but more importantly, focusing in on the pools that are derived. Again, and to be clear, these are bulk selected pools in less than a week that have this incredible uniformity. So what's next? Well, this is what we view as the world of complexity in front of us for the next versions of our vectors and the leap-in transposase platform. We're looking now more and more at 3-chain and 4-chain molecule, the so-called pool of bi and multi-specifics. What's become evident over the last few years and even earlier, of course, last decade, is that manufacturing of these is complicated. Understanding the assembly of these complex molecules is not easy, and varying the amount of the chains as well as the ratio of these chains is something that drives a CHO cell to be able to create the molecule of interest to treat patients in need. So what are the considerations that one needs to take when thinking about chain balancing, when thinking about multiple chain molecules and how to tease apart how much of each chain is the right amount for your platform or for your molecule of interest? I've loosely bucketed this into 2 different categories, sequence-specific considerations and vector. In no way is this meant to be comprehensive. Clearly, these are primary sequence-specific items that can affect expression and hence relative chain ratio. Things like poly-A signal, things like codon choice, all items that squarely focus on our expertise in our gene GPS capabilities. But of course, there's vector configurations as well. Which promoters do we choose? What's the order of the expression cassettes? How many expression cassettes do we put on one vector? Should we have one or multiple vectors? What's the spacing? What's the directionality, etc.? There are many, many complicated variables here, which can be thought of as part of the solution set and feeds very nicely into our, again, 2 decades of experience using that design, build, test, learn methodology and algorithms. Okay, so we applied this to a relatively simple problem first, a two-chain problem. You can think of this as a heavy of light. You can think of it as a knob into hole SCFE type of bispecific molecule, but suffice it to say we have found solutions that work incredibly well for two-chain types of molecules where we want to vary the relative ratios of one open reading frame to another. In this case, 14 different vectors designed to give different expression ratios, and you can see quite elegant control of those expression ratios using the platform. This is a two-chain example. Here's a three-chain example. In this case, 6 different expression constructs where the goal, again, was to vary the relative ratio of each chain to the other chain as a function of the various elements that I referred to inside those expression cassettes, and again, able to find solutions inside that complicated design space that allow us to do exactly that. So with that, what I'd like to do is give you 2 case studies. I'll say up front that these are both case studies of 3-chain bispecifics, where we've worked with our customers to help solve problems for difficult to express molecules in this space. I need to say a priori that there's a lot of data here I'm unable to share because of confidentiality reasons, but I'll try to do the best to make as clear the point of each one of these case studies as possible. This first case study, I cannot tell you what the company is, but it's one of our licensees, a relatively large biotech pharma group who's very good at making biologics, multiple products on the market. And in this case, they're looking at a 3-chain molecule where in blue, I will show the desired properly assembled molecule, and in pink, the assembly variants. In this case, for this molecule number 1, their in-house platform gave the following results. And what you can see is a combination of low titer and about 30% of the molecule being expressed as an assembly variant. We've screened various numbers of these 3-chain vectors, varying chain amounts, varying chain ratios, and that resulted in a leap-in pool with not only a significantly increased titer, but now instead of going from about 65% molecule of interest, we are now able to create a pool that has 90% of the desired product, which itself is at nearly gram per liter range. A more complex situation was brought to us here. An interesting situation, of course. They're getting 1.2 grams per liter, but 90% of it is garbage. 90% is this assembly variant. This is clearly a non-manufacturable situation. By screening vectors, by varying chain ratios and chain amounts, we were able to deliver the following solution where they had 90% of the molecule of interest, itself at a titer of nearly 2.2 grams per liter, clearly enabling the possibility from this pool to create clones for moving forward in their development pathway. So this is just one example with a company where applying these chain balancing as well as chain amounts in vector screening was able to create pools that can subsequently generate clones to enable the manufacturing of products that might not otherwise have been taken to the clinic. So I'll switch gears now to a second case study here. This one done in collaboration with Rentschler Biopharma, also one of our licensees. Here I can give you a little bit more data, which is good, a little bit more satisfying scientifically. Suffice it to say, we were with them, designed 14 vector configurations. These vary not only the expression levels, but also the expression ratios of each of the 3 chains, as I've referred to in the past. They went ahead and created the pools, and then used these pools to look at product quality and titer. I will note that the analytics used by Rentschler at this case cannot distinguish between the 2 heavy chains in this molecule, and so you'll see this presented as a combined heavy chain 1 plus heavy chain 2. And of course, importantly, what percentage of the molecules that are being made are actually the correctly formed bispecific. As I mentioned, there were 14 vectors. We will focus on a subset of those for the interest of brevity and time. And here are 5 of those vectors, again, creatively named A through E. And you can see that each of these were able to deliver a different relative ratio as well as absolute expression level for these molecules. You can see in the relative ratio, this has been normalized to the heavy chain as 1. And you can see here the amount of excess light chain in the presence of a certain amount of heavy chain. from a relative perspective. And then here on this side, you can see, as I mentioned, we weren't just looking at the amount of each chain, but from a relative perspective, but also an absolute perspective. Here, estimated on a micromolar basis, the amount of each chain being expressed by the CHO pools. And you can see this also varies from construct to construct. Again, to iterate, this is a subset of 14. You can imagine the robustness and the richness of data that was generated as part of this project. So here's the titer and the product quality information. As you can see, the titer in pink ranged from about 600 mg per liter to nearly 2 grams per liter, and the amount of properly formed bispecific from roughly 50-50 up to 89 to 91 or so % of the properly formed bispecific. So let's take a look at a subset of these conditions and see what we can see. Here you see two vectors that gave a comparable amount of heavy chain to light chain ratio, but varied a bit, not a lot, but some reproducibly in the amount of each one of these chains expressed. Almost no effect on titer, both of them sitting here in about 900 mg per liter, but just this minor difference in the amount really was able to drive a significant difference in assembly from roughly 50-50 up to nearly 80% of properly formed bispecific. I'd like to point one other pool. Again, comparable ratios, 1.2, 1.3, 1.4 to 1 with respect to the relative amount. However, a significant increase now in the amount of each molecule being expressed, and now we've come into a situation where our titer is now up to 1.9 grams per liter in pools, and our product quality showing 89% roughly properly assembled bispecific. So by virtue of screening these vectors, varying ratios, varying amounts at the pool stage, using LEAP-in where we know that the integrations will be intact, these pools will be predictive of our clones, we can now take these pools, or by we I mean Rentschler, take these pools and really ask the question, what do the derived clones look like? And so they did that from these two pools, which I pointed out earlier, one at 50-50 and about 0.9 gram per liter, and one at about 88-89% bispecific and 1.9 gram per liter. So pool A created the following data. Here the pools were screened in 24 deep well format. First, I'll point out the uniformity of expression, which I mentioned too early. Again, a tribute to the uniformity of these pools that are being generated. Interestingly enough, from a bispecific perspective, when they looked at the clones, selected clones based on titer. They identified clones at nearly 2 grams per liter from this pool, but the product quality was even worse than the pool. You can see more and more of this mispaired antibody in yellow in the derivative clones as they selected for higher and higher titer molecule clones. Pool E, the winner in the clones in the pool selection process. Here's the titers that they got in their 24 deep well format for screening. Again, relatively uniform titers, a hallmark of the pools that we generate. But more importantly, as they selected clones for high titer, notably in this case, 5.5 gram per liter clones, the product quality, the percent of the bispecific antibody at interest was nearly identical from the pool through to the derivative clones. So what this means is that the pools that were poor resulted in high titer clones that were poor or poorer than the pools, but the pool that was the best actually predicted the quality of the clones. And in this case, very high quality, properly assembled bispecific at 5.5 gram per liter in a scalable process. So with that, I'm going to stop the presentation and just remind you of these unique qualities that come out of the derived pools from our platform. They are quite predictive, at least when compared to other methodology, of the clones. Titer, assembly, product quality, etc. This is uniquely enabling for the ability to screen vector configurations, they might actually be confusing, vector configurations at the pool stage, especially for complicated molecules. It's not just the ratio, it's also the amounts, and we see this over and over again. And as we continue to push our R&D in this space, we're starting to see patterns that we're applying to future molecules as well. And of course, the end of the day is to get this to patients. These pools, whether it's just a simple antibody or whether it's a more complex bispecific, do reduce the timeline and the risk to getting into phase 1. You can transfer these pools very early into the manufacturing organization. They can use them for process development and analytical development, using material from the pools. And I've already mentioned there have been one, but there's also a second IND that was filed that was COVID related based on pools. But now we have many of our customers and our licensees looking at using pools for tox lots in parallel with clone selection, MCB, and characterization of clones. So with that, I'll stop, and hopefully I've convinced you that our pools are quite unique. And I'll just say thank you to our partners in this. Our cell line development host cell line is derived from the Horizon Discovery ChoSource cell, a GS null cell. I've also mentioned Rentschler Biopharma, who has done a fantastic job in collaborating with us on some of these complex molecules. And last but certainly not last, our hundreds and thousands of customers that come to us every day to inspire us to innovate and who bring to us some of their toughest problems so we can innovate and solve those solutions for the industry. And with that, I just thank you for your time. Hopefully I haven't gone over, and look forward to any questions you might have. Thank you.